Hello and welcome. My name is Robin Altman. I'm from the biology department at Sac State, and I'm happy to share with you a little bit about the advanced cell biology course that I taught in the spring and how we pivoted when the pandemic disruption hit. So advanced cell bio, it's a laboratory course, upper division. It's capstone-like. We don't really have capstone courses in the biology department at Sac State, but this is pretty much as close as it gets. Enrollment is small, only about 20 students, and they are almost all graduating seniors, graduating biology major seniors. So for this course, our cure generally has two main arms. The first arm has students focusing on mastering practical laboratory skills, so just learning techniques and methods for the care and culture of healthy mammalian cells. They then use those practical skills as well as some background research to develop and execute a small group research project. That research project has sort of four main phases or four main components. In their groups, so students work in groups of 5, they have to conduct a literature search to identify a toxin of interest that they think could be located in the American River and could have adverse effects on mammalian cells. So the American River runs right next to Sac State, so they're very familiar with this waterway. And so they do this research to figure out what toxin they would like to investigate that could potentially be affecting the ecosystem and many of the different species that live in the waterway or enjoy the waterway like humans. So they sample river water and perform mass spec in collaboration with our chemistry department to determine whether or not the toxin is present. And then they design and conduct a high throughput experiment to be able to test their toxin on mammalian cells, these cells that they learned to culture earlier in this semester. And they're looking at cell viability and cell death as their primary outpoints or endpoints. They then use all of these results as their preliminary data to write a full research funding proposal, which they submit at the end of the semester. So at the time of the COVID-19 disruption, students had already identified their toxins of interest and submitted their pre-proposals. They were unfortunately unable to do any of the hands-on work. So they couldn't do their experiments, they couldn't gather preliminary data, they couldn't do any mass spec analyses. So what we tried to do, how we modified the cure, is that we attempted to expand the research funding proposal writing part of the cure. So what usually happens is students are usually restricted to 3 specific assays that they can use for their proposals and that they can do in the lab to gather their preliminary data. This is just based on funding restrictions as well as reagent availability sort of restrictions, as well as the timeline for the course. So instead, what I did was I introduced new opportunities for students to explore different assays that they wouldn't normally have been able to learn about or do any work with in the lab. Right? And this was instructor-led curriculum, so I taught them about these different assays, essentially presented them with sort of a buffet from which they could choose to go forth and investigate specific assays that they thought would be appropriate for investigating their own hypotheses for their own toxin. So students worked with each other in their groups to research the assays I presented to them, decide which ones they wanted to pursue for their proposal, and then with their chosen assays, they expanded their proposal to include an experimental design plan that incorporated these new assays. They also created their own graphs of what they thought, their best guess, their best prediction of preliminary results for these new assays, should their hypothesis be supported, and included all of that in their expanded research funding proposal. So I tried to gather some informal feedback from the students about these modifications, both during class discussions, and I included a feedback question on their final exam. I think it's important to note that none of this was anonymous. Students commented that in the what worked well category, they really liked being able to incorporate new assays, being able to research different assays and expand their proposals. They said that to them, this made writing their proposals be much more authentic, that they could go in different directions and incorporate new relevant assays for their hypothesis and their research question. Across the board, in the what did not work well category, they said that they didn't think their research experience was as authentic or as impactful since they couldn't actually do their experiments in the laboratory, which was understandable and expected. So what do I take away from this? I think either in person or online for this cure, letting students or introducing opportunities for students to incorporate different assays, new assays into their research proposals really merits consideration. The students really seemed to enjoy that and really ran with it, which was really fun to see. This first time around, like I said, it was very instructor heavy, very top down in introducing these new assays that the students could explore. And so in the future, I'd like to shift that to a much more student driven approach, really give them their reins and let them explore, find different assays that they think could be appropriate. I think that would make it much more engaging for them in the future. So with that, thank you very much for your time and attention. And there's my contact information.